which would be agglutination, precipitation, and latex labeled assays. But it's also going to be over the tests that we did in lab. So what tests have we done in lab that we need to consider here? Cold agglutinin, ERF, orgidipine, we did strep. What else have we done? We did mono, H. pylori, flu, what
Or no, it's gonna be with
Did we do any other ones? We did strep, mono, H. pylori, RF, flu, RSV, mycoplasma, and RH plasma or mycopylori. I like mycopylori, yeah. Okay, so these are the tests that we did in lab. But the other thing that goes with these is that we did agglutination reactions. We did EIA. And the EIA we did direct and indirect. So we have these particular organisms and tests, but we also have the methodologies by which we did them. Okay, now the methodologies are covered mostly in the lecture material. If we go through real quickly on these, because you have to know what is provided by the patient, we're just going to put what's provided by the patient for each of these tests, because if the patient provides one thing, then what's in the kit? The other, okay? So strep, is this antigen or antibody? Antigen. Mono.
Patient finds.
The patient is antibody. H. pylori. This was an antibody test that we were doing. Mycoplasma. Flu and RSV. Remember we said if you're trying to figure it out, if you collected a specimen other than blood, that's pretty much going to always be antigen. I guess there's maybe some exceptions out there, but as far as I know, any specimen other than blood is going to be antigen. Cold is looking for antibody, RF is looking for You did the tube titer on the RF? Oh, we did the slide test too. Yeah, it's looking for antigen. On the RF. The RF is antigen. The cold is antibody. Although the antibody there is not specific. It's any cold reacting. Okay. So if you collect something other than blood, you're looking at antigen. That's about as specific as I can always get. Blood, it could be either. Most of the time, if you're collecting blood, it's antibody, but that is only most of the time. That's not a hard and fast. Which agglutination tests did we do? RF and mono. I want you to read well. Yeah, we'll try to get to that shortly here. When I'm looking at agglutination, I'm looking for particle agglutination more on the card. Yeah. The RF we did was agglutination, little strep test, or little agglutination test. The others were all EIA. Which of these was indirect? Which of these was an indirect test? Mycoplasma. Okay. Yeah. Or Myco pylori test. Yeah, I know they didn't work, but the point of the test, the point of that one was to understand that there's multiple steps. Okay. None of these were competitive. These were all direct, or all non-competitive. All right, so let's go to the study questions. Maybe try to narrow it down a little bit, or at least answer through some of these. Where does agglutination's reaction take place?
Surface of the carrier.
On the surface of the carrier particle. And what can we use for a carrier particle? Give me some examples. What do we use? What's our most common thing we use for a carrier particle these days? Latex. Latex particles is most common. It could also be on the surface of a red cell. What are the two key steps in agglutination? Sensitization and lattice formation. Sensitization just means the recognizing of the antigen antibody. And then what forms the lattices? Why do we have lattice formation? What forms it?
They share
They share, be more specific.
the binding site or
Yeah,
the
they have to
constant?
share binding sites, so they have to share the particle. And in order for the V shape of the FAB region, in order for those to bind to more than one, they have to pull them close. Physically, they have to be close enough that the antibody can reach both of them. Yeah, they have to share the particles, because if the antibody is on the particle. That a single immunoglobulin hooks to more than one particle, and that pulls them physically together. And we already talked about that. Well, that's three. The cross links between antigens and antibodies is the lattice formation. Give an example of each of the following types of agglutination. Let's narrow this down a little bit. Direct agglutination, anything we did in lab.
Just maybe test.
Well, a direct agglutination is going to be coated particle, patient, look for clump. We don't have any agglutination inhibition ones. Let's just do direct, then the DAT and the IAT. What's the difference between a direct antiglobulin test and an indirect antiglobulin test? And what antisera do we use in those? Direct antiglobulin, and what section of the lab does those?
EIAs? Or
Again?
EIA?
Nope.
Or
Blood bank does these. What's the difference between the direct antiglobulin and the indirect antiglobulin? It's the same thing that's the difference between any direct and indirect test. direct test,
Okay, add a Y.
pardon?
Don't you add a Y?
Yeah, a lot. It's a multi-step test for the indirect. Okay. In a direct antiglobulin, I am looking for in vivo coating of the cells. So what the patient is providing is antibody-coated cells.
That's what I read, correct?
This is for direct. So when we add our Coombs sera, which is anti-human IgG, that's the antibody that we made to human antibodies. When we add our Coombs sera, we get a reaction. In the indirect antiglobulin test, just like any other indirect test, I am going to take and incubate. It's the sandwich. It's the sandwich. I'm going to incubate the red cells, and I'm looking for in vitro coating of the red cells with antibody. And in that case, what the patient is providing is free-floating antibody in their plasma. So we're looking for antibodies in the patient's plasma. In the direct antiglobulin, I'm looking for antibodies on the patient's cell. Barb will be so impressed if you can remember this all in the blood bank.
And you said you're looking for in vitro coating of the antigen or seed?
In a direct antiglobulin, I am looking for antibodies on the patient's cell. In the indirect antiglobulin, I'm looking for antibodies in the patient's serum.
You said those are free floating, or are they?
They're free-floating, yeah. So when I'm looking at the direct antiglobulin, I'm looking at patient's cells, and I'm looking to see if they are coated with antibody in vivo. In the indirect, I'm looking for patient-produced antibodies against donor cells. I'm doing a sandwich method, and it's all in vitro in the test tube. Okay, so these are free-floating antibodies from the patient, which are going to react with donor cells. And I can think of about 20 different questions I could ask on either of these things, but I have just given you the answer to all of them. And I won't ask 20 on this one subject. I do want to go back. What things can enhance lattice formation? What I really want you to remember on that one is low ionic strength solution, that we can decrease the ionic strength, that's where we use LIS. I think that's the point where that one's most appropriate. Two examples of false negatives and two examples of false positives. Let me help you out here. Tell me what's be able to, and I'm pretty much guaranteeing this is going to be an essay question. Describe prosome.
Too much antibody.
Too much antibody, and why do I not? What does it lead to? A false positive or a false negative?
Negative.
I heard both answers.
False negative.
False negatives. Okay, it leads to false negatives because it overwhelms the test system. So the key things I'd be looking for in an essay question there are too much antibody, false negative, overwhelms the test system. What's postsome?
Too much.
Too much
Antigen.
antigen. And why does that matter? How does having too much antibody or too much antigen give me a false negative reaction? I should have plenty to measure.
Because it's reacting with it instead of the patients. Or the other way around.
It's not healthy. Because they don't share. It's not
Because they don't share. That's the key right there, because they don't share. There's enough that I don't have to share. If I put a group of children in a room and I give them 2 balls, they're all gonna kind of get together so they can play with the 2 balls. Okay? If I give them each their own, they can spread out and do whatever they want because they don't have to share. So we don't have to have shared interaction. Yes.
You're gonna get posted mostly with the Foldy test, correct?
What?
You're gonna get posted mostly with the Foldy test where they didn't put enough antibodies on it.
Like I said, postzone is not nearly as common as is prozone.
What about the false positive? The same thing?
False positives can be caused by scratches on the cards. They can be caused by improperly doing the test.
Run over or carry over?
Pardon?
When that has a false positive, run over or carry
Run over or carry over? Yes. Some of these keys are up, but not all of them. Nope, none of these. Precipitation. The biggest ones we're going to look for with precipitation. Here we're pretty much looking at the gels. We're looking at RID and we're looking at the octoloni. What is precipitation? A soluble antibody, soluble antigen, formed to make an insoluble complex. There's pictures in here. You need to be able to define the terms avidity and affinity. Remember, affinity is that initial attraction, and avidity is the sum of all the attractive forces. And that's kind of your answers to questions number 2 and 3.
Avidity is the sum of the attraction.
Sum of all of the forces that hold it together. And this is where she addresses prozone and postzone. So antigen excess is Posone, and antigen, antibody excess is prozone. And what we're looking for is the zone of equivalence. Nephelometry measures light scatter. I'm not gonna ask any more about it. Measure light scatter. On Monday, I want to start with lab at 8 because I want to do the HIV lab, which is our octolony, and I want to get those little buggers cooking, okay? Because I'd like to be able to look at them on Tuesday and hopefully Wednesday, or on Tuesday, so that we can see them before the test on Wednesday. Because I think seeing this is going to make a lot more sense to you when we do it. The octolony, the gel diffusion test that we're going to do, I think it'll make a lot more sense, this precipitation will make more sense if we do it and see it. Okay? Because just like agglutination, we did it first and then we talked about it. I think it'll be easier for you. What is RID stand for? No, it's not the stuff that kills lice. Yeah.
Radial.
Radial immunodiffusion, okay? And what you need to really remember about that, that's the one that it's a circle. You get a circle around the well. All of this takes place in agar. Okay? Our precipitation reactions that we use take place in agar. And again, this is not, I don't know of an RID procedure that anybody still does. It's just not, it's just been replaced. Okay, there's just too many sources of error. Contaminated agar being one of them. So to calculate the concentration of a radial immunodiffusion method, that's where you measure the ring. You measure the ring, you square the diameter, and you plot it on graph paper. Okay. So the key here is to remember that you square the diameter of the ring. If I ask one question on this, on RID, that'll probably be it. There's not going to be a lot of questions on it. Octolony, which is the other precipitation method that we work at. Okay. And as far as what do you do if it's higher, like we talked about, you do the same thing you do any place else in the lab, you dilute it. That one's just common sense. I will review over the octolony as we are putting it into the wells and stuff when we do the test on Monday. And Jean said she'd be around, and I might let her review it because she's a lot smarter about it than I am. But basically what you're looking for is I put antibody or antigen in the well here. Obviously, if antigen goes here, antibody goes here, if antibody goes here. Okay. So if I'm putting antibody here, I'm going to put antigen out here. If I put antigen out here, I'm going to put antibody here. Got it? Well, what I'm looking for is I'm going to have this diffusing out. If what is in here is the same as what's in here, it's not going to keep going. It's going to stop. And that's going to be my identity. If it's partially the same, part of it's going to keep going, and part of it's not, so that's partial identity. And if it's not the same at all, they're going to cross because they're not going to stop at all. Okay. They're just going to keep diffusing out. Yes?
Is partial, or no? So neither one of those is partial?
No, none of it. This is identity. So I've got my antibody XY here. I've got my antigen XY here and my antigen XY here. As the antibody comes across, it hooks up with this, and it does the same here, so I don't have it keep going. If I didn't have any antibody to X here, this X line would just keep on going because it didn't have anything that stopped it. If I didn't have any antibody to Y here, it would just keep on going because it didn't have anything to stop it. And we will go, like I said, we'll go over that when we actually do it in class, hopefully when you can see it. Electrophoresis, other than knowing that this is a precipitation reaction, Barb went over this in chemistry, right? So I'm not gonna spend a lot of time with it. We are gonna talk more about it when we talk about multiple myeloma, and we'll discuss the findings there. But other than knowing that electrophoresis is a precipitation method, I'm not gonna ask you much more about it. And Western blot is just a form of electrophoresis. Trying to narrow this down a little bit for you guys.
What was that again about Western blot?
It's a type of electrophoresis. Probably going to be a good chunk of the testing, or a good chunk of questions on this. Well, it'll be a good chunk of all of it. I'm going to kind of combine questions 1 and 2 and simply say, be able to differentiate and explain competitive versus non-competitive assays. Again, sounds like a nice essay question. Some things to keep in mind when we're doing that. Competitive, non-competitive. My color change or my reaction at the end, because I can have a competitive agglutination too if I wanted to. So, but in a competitive assay, if I have, okay, we're going to use color just because it's easiest. If I have a really dark color at the end of a competitive assay, what does that mean? Does that mean my result, my patient result is high, or my patient result is low?
Low.
Means my patient result is low. If I have a weak reaction, then it means that my patient result is
Low.
my patient result is high. They are inversely proportional. Why? Right?
And so if the patient has a low value, then that means that there's more enzymes binding. So you get a higher color change. But if there's a high patient, you get less color change because there's less enzymes binding.
Right, enzyme labeled one's binding. Okay. Everybody get that?
No.
It's an inverse in a competitive assay. The color change, or whatever you're reading, is inversely proportional to the patient's result. Because when you are in a competitive assay, the enzyme or the marked, the labeled thing that's going to show up is on what you're adding from the kit. So if the patient has a lot of what you're measuring for, it will be able to bind a lot of the sites, which means that our labeled stuff that we add will not be able to hook up to many of them, so I'm going to get a low reading. If the patient doesn't have very much of what I'm measuring, it won't hook to very many sites, which means the stuff that came out of the kit has lots of room to hook onto, and that's where my color or my reaction comes from. So since there's lots of room for those to bind, I'm going to get a high reaction. So it's inversely proportional in a competitive assay. So if I have a competitive assay, high result is going to mean low patient. Okay, it means the patient has a low titer, a low amount. A low result or a lack of color is going to mean that the patient has a high result. And in a non-competitive assay, it's the other way around.
What are some examples of tests, specific tests for this?
For a competitive assay, I don't have one that we do here. The cluster, clustering? No, not
Is
clustering.
that what you're using, the OP?
The OMP, the coccidio, or the cryptococcus, not cryptococcus, cryptosporidium, I'll get there. Cryptosporidium and giardia, okay? What you do and what you're doing when you're doing a competitive assay, I'm going to have my well, have a little test tube, okay? And I'm going to take conjugate, which is labeled antigen, because I'm testing for antigens in this test. It could just as easily be antibody, right? And I'm going to put patient antigen, and I'm going to put them both in at the same time. And there are little binding sites. And so the conjugate labeled ones are going to try and get some. The patient ones are going to be, trying to get them. So the more I have the patient, the less of the conjugate that we'll be able to get on there. In a non-competitive, what I have is I'm going to put my patient in, let it do its thing, and then when I'm finished, I'm going to put my conjugate in. Okay? But in a competitive assay, I'm putting them both in at the same time. Let them duke it out.
But non-competitive is with the anti-human, where it goes on.
It can be either. And yeah, it can be either. I can have a competitive where I use Kumsira, where I will put in a labeled conjugate and then use Kumsira to get my reaction. Okay, but then I would label my conjugate and I wouldn't be labeling my, let me see if I can show you what that would look like. That would be an indirect. So on indirect, we've got our subs, we've got our well slide, whatever it is. What I would do here is I'm going to put in patient antibodies, okay? And I'm going to put in labeled antibodies to whatever this is, okay? Then I'm going to put in Kumsira, because that's what's going to give me my reaction. And it's going to react with these, but it's still, my color reaction is going to come from the conjugated antibodies that I put in at the same time I put in patient antibody. And I may not label this, I could actually put in, horse antibody to combine with it. And then when I put in the labeled Coombs, it's only going to attach to the human. Okay, that's another way we can do that competitive. Okay? Does that make more sense? So this would be an indirect. The one that I showed you the first time would be a direct. So there are, like I said, you can have a direct or indirect, competitive or non-competitive. Just depends on what I put in. In the competitive one, the main thing to remember is we put them both in at the same time. In the indirect, we put one in at a time. We can also do one where what I do is I put in the patient, and instead of putting in an indirect, instead of putting in an anti-human globulin label, what I can do is I'll put in the patient and let it react, wash away any excess, then I'll put in a labeled let's say we were looking for rheumatoid factor. I could put in the patient and let it react first, and then I could put in an anti-rheumatoid factor monoclonal antibody to react. I could put in a labeled one, and that would be a direct, non-competitive assay. Make sense? So I could put the patient in, let it go first, wash off any extra, and instead of adding a Coombs to make the sandwich technique, I could just add a tagged monoclonal that would then go and attach to any sites that were left. Okay. So there's a numerous ways we can mix and match these things. It just kind of depends. This is what they do in research labs to figure out, you know, how to make it tech-proof. And somebody works in a research lab, and she was always like, "Oh, we got to make it tech-proof for those people who don't, you know, follow the rules, because we know they're gonna take shortcuts." So, you know. All right, what is the difference? Let's see. All right, we've already talked about competitive versus non-competitive, and like I said, honestly, that sounds like a very high point essay question, because really, that's a big chunk. Okay.
So non-competitive, is that proportional then?
Non-competitive is proportional, yes. So if I have a high color, then that means I have a high amount in my patient. If I have a low color, I have a low amount. It's like the mono that with, you know, the little strip that you stick in there. We've actually seen patients that as it comes up the paper, it turns the test strip dark blue before it even manages to get to the control line. And you're like, well, that person's got a massive case of mono because it comes up so quickly. Or we did have one H. pylori that we did that with. It hit the test line, and H. pylori is notorious for weak, wimpy, wussy reactions. You have to look really closely. This thing hit that test line, and it was like, bam. It didn't even make it to the control line before it was super positive.
You can't read that out though
Until the control line comes up, yeah. Well, we just waited the few seconds till it hit the control line. We didn't even set the 10-minute timer. We're like, yeah, we're done. Once it's positive, it's positive. All right. What is the difference between a heterogeneous and a homogeneous enzyme assay? There we go. We've talked about competitive, we've talked about non-competitive. Capture assay is the sandwich assay. Oh, okay. In the heterogeneous ones, the enzyme labeled reagent doesn't react in the initial binding. Okay. In the homogeneous enzyme in a homogeneous enzyme immunoassay, any antigen antibody system where we don't have a separation step. Okay? The heterogeneous would basically, it's going to be a two-step of some sort. It may not be a sandwich technique, but it's going to be a two-step of some sort. And honestly, I don't know what two viruses are commonly detected with the capture assay, because we use it for a crap ton of stuff. So I don't know which two she's looking for here, so obviously that's not going to be a question.
It says roto and RSV.
Okay, rotavirus and RSV, that's nice. But we also use it for like a half a dozen others. It also can be used, we have a flu kit that does it. We also have a couple of other ones that do it. So I would say that that question is a little off. It can be used, any of these methodologies can be used for pretty much any kind of testing. What is used as a fluorescent tag or label in the direct and indirect FIA or IFA methods?
Forcing.
Fluorescein. And you are going to want to know that one. And the IFA method that we're going to do an ANA. We're going to look at those next week. We're going to do an ANA. And an FIA would be fluorescent immunoassay, and an IFA would be indirect fluorescent assay. Okay, so we have a direct and an indirect, just like anything else.
So FIA.
Is a fluorescent immunoassay. And an IFA is indirect fluorescent assay.
When you said A and A,
ANA
you
is the
mean
test.
A and A or A N?
Oh, ANA, three initials. Anna. Okay. Direct assay, we use this one for treponema. We use this one for, I don't remember what other ones we do. This one says chlamydia. But basically in this one, what we're gonna do is we're gonna put, this one here would also be what we do for when we talked about our our immunohistochemistry things, except that instead of fluorescein, it's gonna be tagged with an enzyme or with a stain. Okay. So direct FIA, indirect is going to be patient antibody wash away, add a flag. Okay. Radioimmunoassay, why don't we do it anymore? That's kind of the big piece.
Sensitivity.
No, it's really sensitive.
Difficult to get rid of the byproducts.
Yeah, well, yeah, it's dangerous. They don't like us working with radioactive material. They're safer. Easier to deal with. I mean, it doesn't seem like a lot of material when you just look at what PowerPoints and stuff, but this is a huge piece of serology. And why is serology important? You have to remember, before the advent of being able to use antibody reactions, we either had to grow it, which many things don't grow, viruses don't grow. Viral cultures are horrid. They don't do much for us. All right, so we have a choice here. We'll review again on Monday, go over any questions you might have. I'll have written the test by Monday, so I'll make sure that I've covered everything that's on the test. We can either go on and do the next lecture, or we can go to lab for a while and I'd like you and redo the test. But like I said, when you are doing them, what I want you to do, we're gonna pull names from a hat, I'm gonna pair you up with somebody, and I want you to do them together. And I want you to explain to each other what is going on at each step when you're doing the test. So when you're doing the agglutination, you're gonna talk about sensitization, you're gonna talk about lattice formation, you're gonna talk about why is it positive, why is it negative, which one's susceptible to prozone. So when you're doing them, I want you to be studying at the same time. Talking about when we do, I can pull the mycoplasma kit out and we can do mycoplasmas again, so that, one per desk is fine, we'll do that so we don't waste too much reagent, but I want you to do each of the different test methodologies and discuss whether it's a direct or indirect, and how you know, and what's happening at each stage, and what you're putting on there, okay? thinking about what you're actually doing and not just 1, 2, look for blue. So do you want to go on to the next lecture thing? Because if we don't, we're going to be really pushing lecture next week.
I mean, we do have to.
We gotta get through it.
Yeah, we've got to get through it. So what we're going over today is not on Wednesday's test. So what we're going over today is not on Wednesday's test. And since we only have 2 tests left, what I am going to do is, like I said, we'll look and see what we get through. I may have a take-home, and you guys seem open to the idea of a take-home, that you'd like that better, instead of a quiz in class. So if we don't get to everything on Monday's test, then what we will do is I'll give you a little take-home on for a Wednesday. Or I might give you a take-home, just kind of a overall kind of thing. If I feel like there's some sections you didn't do so well on, I may give you a take-home test just for some bonus points. Okay? We will see how things play out here. And molecular diagnostics, I will tell you,